Hello, my name is Andrew Joseph, and I'm going to be presenting to you today my undergraduate research project on the evaluation of isobavachalcone, also known as IBC, as a lifespan extending compound in C. elegans. We'll start with a little bit of background first. In this experiment, we focused on lifespan, which is the total amount of time an organism lives, and there are two important types that we focused on. Maximum lifespan, which is the longest any individual of a species can live, and average lifespan, which is the typical age individuals reach in a population. Lifespan is determined by biological processes such as aging. A key driver of aging is going to be oxidative stress, which is defined as an accumulation of reactive oxygen species, which can cause damage to DNA, proteins, and lipids. Now, organisms can counteract this damage using conserved stress response pathways that influence longevity, which we'll talk about a little bit later. Certain bioactive compounds, such as antioxidants, can alter and affect those pathways. By neutralizing oxidative stress, they can potentially slow aspects of aging and overall promote longevity. In this experiment, we explored two different antioxidant compounds. The first one being resveratrol, which is a compound commonly found in red wine. This compound is a well-established lifespan modulating compound known to activate stress response and longevity pathways, which include those DAF16 and FOXO transcription factors, which we'll talk about a little bit later. Because resveratrol is known to promote longevity, we used it as a positive control to validate the lifespan assay conducted in this experiment. The second antioxidant compound we explored was isobavachalcone, also known as IBC. This compound is a compound that is commonly found in green tea. It is a bioactive flavonoid that has antioxidant and metabolic effects. And we see this as a compound that has the potential to influence similar aging-related pathways as resveratrol, but effects on lifespan are not clearly defined. So in this experiment, we'll go ahead and evaluate lifespan modulating potential of this compound. To test these compounds, we use the model organism C. elegans, the wild type N2 strain. We use this organism because they are very small in size, have a very short lifespan lasting around 2 to 3 weeks. They're very simple, well-mapped organisms with every cell lineage known and researched, and they're very easy and cheap to grow in large numbers. The stages of development for C. elegans was very important in this experiment, as we had to go ahead and make sure that the ages of the worms were synchronized in order to accurately determine the effects of the compounds on lifespan. Now, aging in C. elegans is strongly controlled by a signaling pathway that is very similar to humans, known as the insulin and IGF-1 pathway. When insulin is released after eating, it tells the cells that energy is available. When that happens, it activates a chain of proteins inside the cell, including AKT1 and AKT2. These AKT proteins go ahead and phosphorylate these transcription factors known as DAF16 in C. elegans and FOXO in humans. When these transcription factors are phosphorylated, they are prevented from entering the nucleus. And when that happens, the cell focuses more on growth and less on stress protection and repair. For this experiment, we propose that IBC will inhibit and reduce the insulin IGF-1 signaling, which will decrease the AKT activity and allow the DAF16 and FOXO homolog transcription factors to enter the nucleus. When these transcription factors enter the nucleus, it promotes stress resistance, the enhancement of detoxification of reactive oxygen species, an increase in survival under stress, as well as cellular repair mechanisms. Now, IBC is not very well characterized in terms of its effects on aging and lifespan, but it has been reported to exhibit antioxidant activity, which is relevant because oxidative stress is a key factor in aging. Because of this gap in knowledge, the central research question for this experiment asks, does IBC extend lifespan and promote longevity in C. elegans, and how does its effects compare to resveratrol? The hypothesis for this experiment is if IBC functions as a bioactive antioxidant, then it will increase lifespan and promote longevity in C. elegans by reducing oxidative stress and modulating those conserved longevity pathways that we spoke about. Now for the methods part of this experiment. We begin with the drug preparation, including the compound FUDR. This is a compound that is commonly used in C. elegans lifespan assays as a sterilization compound to prevent the worms from reproducing and having any progeny. We also prepared IBC and resveratrol to be added into the agar to later test the effects. For the worms, we also prepared an E. coli OP50 bacterial culture and used that as the food source for the worms. In order for the worms to grow and to age, they have to have a food source that will keep them sustained throughout the time frame of the experiment. And we do have an experimental protocol workflow that will explain this a little bit better. For the drug preparation of this experiment, we went ahead and added the drugs to the NGM light agar at 55 degrees Celsius before plating them and allowing them to dry. After that, we went ahead and seeded them with the bacteria OP50 culture that we grew. And then we had to maintain the populations of worms. We did this by transferring a small agar chunk onto the fresh OP50 seeded plate and allowing those populations to grow for a week before using them for the age synchronization process. The temperature of the incubation for this experiment was very important, as the temperature controls the aging speed of the worms. When we have higher temperatures, we have a faster metabolism in the worms, which allows them to age and die sooner. If we have a lower temperature, it causes the worms to have a longer lifespan. In order to see the worms, we had to use a microscope as well as polarizing film, which improved the visibility and contrast. Being able to accurately see the worms and count them was a very important aspect for determining lifespan and the effects of those compounds on lifespan. A major part of this experiment was the worm synchronization procedure. As we mentioned before, having the worms synchronized in their age allows us to accurately determine the effects of the compounds that we use in this experiment. After about 6 to 7 days of growing the worm cultures, we then have enough of egg-carrying adults on those worm plates to wash them off and collect them and use them for the synchronization bleaching process that we'll talk about. Now, there is an experimental workflow that goes a little bit more in depth with this, but essentially we go ahead and collect the worms using a M9 buffer on those plates that we grew up. Once we have all of those worms collected, we use a bleach solution to go ahead and dissolve all of the adult worm bodies, which leaves behind the embryos and the eggs. The embryos in those eggs are protected by a tough eggshell that resists chemical degradation, whereas the adult tissues are rapidly degraded due to that bleach solution. What's left behind after we go ahead and use those solutions and spin them down are the eggs, which are left to be incubated in a solution of M9 for about 24 hours without food, which goes ahead and causes them to enter an L1 arrest, where they enter that L1 stage of development and stay there because they have no food to grow into the later stages of development. Once we have all of those worms synchronized, we go ahead and transfer them to the NGM plates and allow them to develop at 22 degrees Celsius before picking them onto their respective condition plates. As you can see here, this is the experimental conditions that we use with the control, the resveratrol, and the IBC of varying concentrations. We used about 10 to 14 worms per plate with about 3 plates per condition, creating a technical triplicate. And we did 2 independent trials this semester. We did the trial 1, which was the control versus the resveratrol, and trial 2, which is the control versus the IBC versus that resveratrol. Now, for the lifespan assay and lifespan measurement, the young adults were transferred to treatment plates were considered day 1 for the lifespan assay, and we scored those worms about every 1 to 3 days. The live worms were considered worms to have movement, pharyngeal pumping, and the dead worms had no response to any gentle touch to the pick, and they were subsequently removed after counting if they were dead. Now, the experimental workflow, as you can see here, kind of explains the methodology a little bit better. You can see we go ahead and take those drugs and mix them with that agar to get the desired concentration from previous literature and previous studies that have been used. We go ahead and pour those onto the plates where we allow them to dry, and then we take that OP50 E. coli culture and add that to those plates, and then have those plates dry in the incubator as well. After that, we go ahead and synchronize the worm age populations. As we mentioned before, we go ahead and take all of those worm plates with that M9 buffer, use that bleach solution to get the eggs. And once we get those eggs, they hatch and enter an L1 arrest because they don't have any food. And once they don't have any food and they enter that L1 arrest, we go ahead and plate them onto an agar plate where they are able to be picked and transferred to their prospective treatment conditions. Now for trial one, you can see that the control had a maximum lifespan of 21 days, whereas resveratrol was 23. And the interpretation is resveratrol has about a 2-day increase in maximum survival under the experimental conditions. Average lifespan you can see for the control is 13.23 days, whereas the resveratrol was about 14.32 days. The interpretation here is that resveratrol extends average lifespan by about 1 day under these experimental conditions. Survival percentage did decrease in both groups over time, but occurred more rapidly in the control. The control group declined quickly and reached 0% by day 21, whereas the resveratrol group survived longer, maintaining worms later and reaching 0% by day 22. For this experiment, we used the Kaplan-Meier curve to accurately visualize the lifespan of the worms. As you can see here, the resveratrol survival curve shows a rightward shift and remains higher than the control during later portion of the experiment, which does indicate an improved late life survival. The curves show late separation without crossing, which suggests that the consistent survival advantage for the resveratrol treated worms was present. For trial 2, we can see the maximum lifespan of the control was 26 days, resveratrol was 27 days, IBC 0.6 was 26 days, and IBC 0.2 was 27 days. The interpretation is that resveratrol does slightly increase maximum lifespan by about 1 day. Similarly, IBC 0.2 does the exact same with a maximum lifespan increase by about 1 day, but IBC 0.6 does not improve maximum lifespan with the results similar to the control. The average lifespan we can see for the control was about 13.66 days, resveratrol was 18.29 days, IBC 0.6 was 14.43 days, and IBC 0.2 was 14.86 days. The interpretation we see here is that resveratrol does extend the average lifespan by about 4 days, and the IBC 0.6 concentration shows little to no improvement versus that control, whereas the IBC 0.2 concentration showed a slight increase by about 1 day. Their survival percentages did decrease in all groups over the course of the experiment and varied by treatment. The control group declined fastest in their survival percentage. The resveratrol group showed an improved survival compared to that control. The IBC of the higher concentration exhibited an intermediate protection, whereas the IBC of the lower concentration showed a similar but slightly improved trend compared to that of the 0.6. For the Kaplan-Meier curve here, we can see the overall trend of the control being that it declines fastest, acting as a normal aging baseline. The resveratrol, which is in red, we can see a clear rightward shift and a higher survival probability compared to that control. The IBC treatments, which are in pink and brown, show mild dose-dependent effects with partial separation curves, but had an overlap with the control later on, which indicates a weaker lifespan extension. Several experimental limitations were encountered during this experiment, including material delays, agar contamination, and synchronization issues. An important limitation to note is the limited statistical analysis that was conducted during this experiment. The number of successful trials in replicates was too low for statistical testing, so more independent trials would be needed to confirm whether the observed differences that we observed were statistically significant. Another important limitation was for trial 1, we did miss a scoring period of about 1 week due to spring break. And then for trial number 2, the FUDR was excluded from the control plates and had to be added on later on in the experiment. Several conclusions can be drawn from this experiment and the results that were obtained from this. As we saw, the resveratrol positive control did successfully increase survival in each trial that was ran, which validated the results and the experimental design and methodology for the lifespan assay. The positive control did have a stronger effect on survival with a higher late-stage survival relative to both the IBC and the control groups. The IBC treatment we did see produce a small increase in lifespan compared to that control group. We also see that it does appear to have a small protective effect under the experimental conditions that we used. The average lifespan in the control group did range from approximately 13.2 to 13.7 days, while the worms treated with IBC had mean lifespans of about 14.4 days for the 0.6 concentration and about 14.9 days for the 0.2 concentration. The slight lifespan extension that was observed in the IBC would indicate a partial activation of that DAF16 FOXO transcription factor homologs that we were talking about, which can lead to longevity response. The slightly greater lifespan that was observed at the lower concentration compared to that higher concentration of IBC does suggest that a possible dose-dependent response was taking place. Future studies, however, would need to explore a wider range of IBC concentrations and increase the replicate numbers and further investigate whether those effects are dose-dependent or mediated through that AKT-16 FOXO signaling. Of course, there would need to be more trials that were ran as well in order to statistically determine whether the results that were obtained were significant. And these are my acknowledgments and my references. Thank you.